The geothermal springs of Taupo's volcanic zone are a mix of breathtaking beauty and scalding heat. The hottest of these springs boil at over 90 degrees Celsius, while the acidity has been recorded as more acidic than stomach acid. And yet, despite these limitations, these springs contain abundant life. One such life form, and the focus of my project, is the bacteria Acidithiobacillus caldus. Its name describes its lifestyle, acida referring to the acidic conditions in which it grows. Theo is a Latin term which refers to sulfur, its energy source. It essentially eats it for getting energy. Bacillus is a term used by microbiologists to describe a rod-shaped organism, like you can see on the slides here. While caldus means warmth-loving, referring to its ability to survive in temperatures as high as 55 degrees. For context, in 55-degree water, you would get third-degree burns after only 30 seconds. Acidithiobacillus caldus is found unaffected by the blistering heat in hot springs and coal mines around the world, especially in New Zealand, where they've been shown to be the second most common bacteria in our geothermal springs. A recent study confirmed this abundance, but they also had some pretty unexpected results while they were at it. They found the DNA of Acidithiobacillus caldus and 3 new species of the same genus in hot springs with temperatures as high as 90 degrees Celsius. 90 degrees! That's a whole 40 degrees higher than decades of literature have even said is possible. In fact, the hottest bacteria in acidic conditions have ever been known to grow is only 80 degrees Celsius. This has obviously raised a lot of questions. Was it just the DNA of a long-dead bacterial cell, or did it come from a cell that was alive, reproducing and thriving, all at 90 degrees Celsius? Are there really 3 new species to find right under our noses? This year, I'm going to answer all of these questions. So far, I've collected samples from the Taupo volcanic zone by carefully extending a 3-meter pole into the scalding depths. Then I put these samples at 45 degrees and 70 degrees to try and get as many bacteria growing as I can. I actually already have over a dozen bacteria from the 45-degree condition, one of which I've already identified to be Acidithiobacillus. That's actually the one you can see right next to me on the slide. Next, I'm going to run a series of tests on my bacteria, and I'll see what they can do. I'll figure out their pH, their chemistry, their appearance, and especially temperature. That way, I can find out once and for all, can bacteria really grow 10 degrees higher than we've ever shown them to grow before? How hot is too hot? Thank you so much for listening.